So here's something kind of wonderful to think about as you're lying there tonight. You are, right now, in this very moment, doing the thing we're going to talk about. You're settling into it, maybe already feeling the edges of it pulling at you. And yet, if I asked you to explain what's actually happening, like really explain it, the machinery of it, the biology, the deep why of it, you'd probably find yourself reaching for words and coming up short. And that's okay, because for most of human history, nobody could explain it. Not the philosophers, not the doctors, not the scientists. Sleep was this enormous blind spot, this thing every single person on the planet did, every single night. And yet, it remained one of the most stubborn mysteries in all of biology. Think about that for a second. We figured out how to split the atom. We mapped the human genome. We sent people to the moon and brought them back alive. And for the longest time, we couldn't answer the most basic question about something we spend a third of our lives doing. Why do we sleep? What is sleep actually for? What's happening in there when the lights go out? So tonight, let's just walk through it, all of it, slowly. How sleep works, from the very first clues that ancient thinkers noticed, through the breakthroughs and the wrong turns and the genuinely wild discoveries, all the way up to what we understand now, which, honestly, is a lot more than most people realize, and still not nearly enough. And if you fall asleep somewhere along the way, well, that's kind of perfect, isn't it? You'll be doing the very thing we're talking about. Let's start way back. And I mean way back. The ancient Greeks thought about sleep constantly, which makes sense when you realize how much of Greek philosophy was built around the question of consciousness, what it means to be aware, to perceive, to think. And sleep was the nightly interruption of all of that. It was the daily little death. In fact, the Greeks personified sleep as Hypnos, a god, the twin brother of Thanatos, who was death. They were literally twins in the mythology, sleep and death, born from the same mother, Nyx, who was the goddess of night. And there's something poetic about that, but there's also something revealing. It tells you that even thousands of years ago, people noticed the deep resemblance between sleep and death, the stillness, the closed eyes, the withdrawal from the world, and they were unsettled by it. Aristotle, around 350 BC, actually wrote a whole treatise on sleep. It's called On Sleep and Sleeplessness, and it's one of the earliest attempts to explain sleep in what you might call physiological terms rather than purely mythological ones. His theory was that sleep was caused by the evaporation of food during digestion. He thought that when you ate, warm vapors rose from the stomach to the head, and when those vapors cooled and condensed, they caused heaviness and drowsiness, and you fell asleep. It's wrong, obviously. Completely wrong. But here's what's interesting about it. Aristotle was trying to locate sleep in the body. He was saying, this isn't just the gods pulling a curtain over your mind. This is a physical process with a physical cause. And that instinct, that impulse to look for a mechanism, was genuinely ahead of its time. He also noticed something important. He observed that sleep seemed to be universal among animals, not just humans, not just mammals, but seemingly everything with a complex body. Birds slept, fish seemed to sleep, even insects appeared to have periods of rest. And that observation, which seems so simple, actually contains a profound clue. If sleep is universal, if evolution has preserved it across hundreds of millions of years in countless different species, then it must be doing something essential. Because evolution is ruthless. It doesn't keep things around for no reason. Sleep makes animals vulnerable. It takes them offline. A sleeping animal can't eat, can't mate, can't watch for predators. From a pure survival standpoint, sleep looks like a terrible idea. And yet, every complex organism does it, which means whatever sleep is doing, it's so important that it's worth all that risk. But nobody could figure out what that thing was. Not for a very, very long time. For most of recorded history, sleep was treated as a passive state. It was the absence of wakefulness, the way darkness is the absence of light. You were either awake and doing things, or you were asleep and doing nothing. That was the assumption, and it shaped how people thought about sleep for centuries. If sleep was just the brain shutting down, then there wasn't much to study, was there? It was like trying to study a television that's been turned off. This assumption persisted well into the modern era. Even in the 1800s, most scientists and physicians thought of sleep as a simple withdrawal of stimulation. The idea was that as sensory input decreased, as the world got dark and quiet, the brain just sort of powered down, like a machine running out of steam. Some researchers proposed that sleep was caused by reduced blood flow to the brain. Others thought it was caused by the accumulation of some kind of toxic substance during waking hours, a poison that built up and made you drowsy, and that sleep was the body's way of clearing it out. That second idea, as it turns out, was much closer to the truth than anyone realized at the time. But we'll come back to that. The real revolution in sleep science, the moment everything changed, came from a technology. And this is a pattern you see over and over in the history of science. You can't understand what you can't measure, and you can't measure what you don't have tools for. The tool that cracked sleep wide open was the electroencephalogram, the EEG. And the story of how it came to be is one of those wonderful, slightly unlikely tales that science is full of. The man's name was Hans Berger. He was a German psychiatrist working at the University of Jena in the early 20th century, and he was, by most accounts, an unlikely revolutionary. He was quiet, methodical, a bit of a loner. His colleagues considered him competent but unremarkable, and he had this obsession that most of them thought was, frankly, a little embarrassing. He believed that the brain produced electrical activity that could be measured from outside the skull. He believed you could, essentially, listen to the brain thinking. Now, the idea that the brain used electricity wasn't entirely new. Richard Caton, a British physician, had detected electrical signals from the exposed brains of rabbits and monkeys back in 1875. But Caton's work required direct contact with the brain tissue. What Berger wanted to do was measure those signals through the intact skull, through skin and bone, in a living, conscious human being. And most people thought that was impossible. The signals would be too faint, the interference too great. It was like trying to hear someone whispering in the next room during a thunderstorm. But Berger was stubborn. And here's the thing about why he was so driven. There's a personal story behind it that's genuinely fascinating. As a young man, Berger had been serving in the German military, and one day during a training exercise, he was thrown from his horse directly into the path of a horse-drawn artillery cannon. He nearly died. At that exact moment, and Berger was very specific about this, his sister, who was miles away, was suddenly overcome with a feeling that something terrible had happened to him. She convinced their father to send a telegram asking if Hans was all right. It was the only time the family had ever sent such a message. This experience haunted Berger for the rest of his life. He became convinced that the brain must emit some form of energy, some signal that could travel beyond the skull, maybe even beyond the body. He wanted to find that energy. Now, he never did find evidence of telepathy, obviously, but in searching for it, he found something arguably more important. He found brain waves. It took him years. He started his experiments around 1920, working mostly in secret because he was embarrassed about the telepathy angle and worried about his reputation. He tried different electrode configurations, different amplifiers, different recording methods. He worked on patients who had skull defects from surgery, where the bone was thinner or absent, hoping that would make the signals easier to detect. And finally, in 1924, he got his first clear recording of electrical activity from a human brain. He saw a rhythmic pattern, a wave that oscillated about 10 times per second, and he called it the alpha wave. He published his findings in 1929 in a paper titled Über das Elektrencephalogramm des Menschen, on the electroencephalogram of man, and the response was almost nothing. The scientific community largely ignored him. Some people were skeptical, others just didn't understand the significance. Berger kept publishing, paper after paper, refining his technique, documenting different types of brainwaves, and still the world mostly shrugged. It wasn't until 1934, when two prominent British physiologists, Edgar Adrian and Brian Matthews at Cambridge, independently confirmed Berger's findings, that the scientific community finally sat up and paid attention. Adrian, who had already won a Nobel Prize for his work on nerve function, essentially vouched for Berger's work, and suddenly, the EEG was legitimate. Suddenly, everyone wanted one. And here's where sleep comes in. Because once you could measure the brain's electrical activity, one of the most obvious things to do was measure it while someone was sleeping. And when researchers did that, when they hooked people up to EEGs and watched the tracings all night long, what they found demolished the old assumption that sleep was just the brain turning off. The brain wasn't turning off at all. It was doing something complex, something structured, something that changed throughout the night in ways that nobody had predicted. The electrical patterns during sleep were completely different from wakefulness. Yes, but they weren't flat. They weren't silent. They were rich and varied, and they followed a pattern, a cycle, that repeated itself over and over through the night. This was the first great revelation of modern sleep science. Sleep is not a passive state. Sleep is an active process. The brain, during sleep, is busy. It's doing work. Different work than it does when you're awake, but work nonetheless. And the nature of that work, well, that's what we're going to spend a lot of time on tonight. The key early discoveries came in the 1930s and 1940s as EEG technology improved and more researchers started doing overnight recordings. A scientist named Alfred Loomis, who was actually a wealthy amateur, he'd made a fortune on Wall Street and then essentially retired to do science, set up a sleep laboratory in his mansion in Tuxedo Park, New York. And Loomis, along with his colleagues, was one of the first to systematically categorize the different stages of sleep based on their EEG patterns. What Loomis found was that as a person fell asleep, the brain's electrical activity changed in a predictable sequence. When you're awake and alert, the EEG shows fast, low-amplitude waves, lots of activity, lots of different neurons firing at different times, a busy and somewhat chaotic signal. But as you start to relax and get drowsy, those fast waves begin to slow down. The alpha waves that Berger discovered, those 10 cycle per second rhythms, become more prominent. Your brain is settling, synchronizing, like an orchestra tuning up before a performance. And then, As you cross the threshold into actual sleep, the pattern shifts again. The waves get slower and bigger. You start to see these distinctive bursts called sleep spindles, brief flurries of activity that look like little spindle shapes on the EEG tracing, lasting maybe a second or two. You see something called K-complexes, these sharp, high-amplitude waves that seem to pop up in response to sudden sounds or other stimuli, almost like the brain is briefly checking in with the outside world and then deciding, no, it's fine, go back to sleep. And then, as sleep deepens further, the waves get slower still, much slower. These are the delta waves, big rolling oscillations that cycle only about once or twice per second. This is deep sleep, what we now call slow wave sleep, and it's one of the most important phases of the entire night. During slow wave sleep, huge populations of neurons are firing in synchrony, millions of them pulsing together in these slow, powerful rhythms. It's like the difference between a crowd of people all talking at once and that same crowd chanting together in unison. And this synchronized activity, this slow, deep pulse, turns out to be doing something remarkable, something we didn't fully understand until much more recently, something that connects back to that old 19th century idea about toxic substances building up during the day. But before we get to that, there's another discovery to talk about, one that came along in 1953 and changed everything all over again. It happened in a lab at the University of Chicago, and it involved a graduate student, his 8-year-old son, and a broken piece of equipment that almost got thrown in the trash. The graduate student's name was Eugene Oserinsky, and his advisor was a man named Nathaniel Kleitman, who was already one of the most important sleep researchers in the world. Kleitman had literally written the book on sleep, a massive tome called Sleep and Wakefulness, published in 1939, and he'd spent decades studying the physiology of sleep cycles. But even Kleitman didn't know what Oserinsky was about to find. What happened was this. Oserinsky was using an old EEG machine to record the eye movements of sleeping infants, trying to study the slow, rolling eye movements that happen as people fall asleep. The machine was old and temperamental, and one night, the pen started going wild during a recording session with his son Armand, who was sleeping in the lab. The pens were swinging back and forth rapidly, tracing huge deflections that looked like the eyes were darting around frantically under the closed lids. Oserinsky's first thought was that the machine was broken. It had to be an artifact, some kind of electrical interference, because the subject was clearly asleep, deeply asleep, and yet the recording suggested the eyes were moving as rapidly as if the person were wide awake and looking around a room. That just didn't make sense. Why would someone who was sound asleep have eyes darting around like they were watching a tennis match? So Oserinsky did what any good researcher would do. He assumed the equipment was malfunctioning. He checked the connections. He fiddled with the electrodes. He tested the machine during waking hours, and it seemed fine. And then, the next time he watched a sleeping subject, the same thing happened. After a period of quiet, deep sleep, the pens would suddenly start swinging again, tracing those rapid, frantic eye movements. And here's what made it truly strange. When Oserinsky looked at the EEG channels recording brain activity at the same time, the brain waves during these episodes didn't look like deep sleep at all. They looked fast and desynchronized, almost like the brain waves of someone who was awake. But the person was asleep, definitely asleep. You could see them lying there, eyes closed, body relaxed, not responding to the environment. And yet their brain was lighting up like a city at night, and their eyes were racing back and forth behind their eyelids, Osirinsky brought this to Kleitman, and to his credit, Kleitman didn't dismiss it. He was skeptical. He was always skeptical. That was part of what made him a good scientist. But he was willing to investigate. They ran more subjects. They refined the recordings. They woke people up during these episodes and asked them what was happening. And this is where the discovery went from curious to genuinely astonishing. Because almost every time they woke someone during one of these rapid eye movement periods, the person reported vivid, elaborate dreams. Not the vague, fragmentary thoughts that people sometimes describe when woken from other stages of sleep, but full-blown narrative dreams, stories with characters and settings and emotional arcs. The kind of dreams you remember in the morning and can't quite shake. In 1953, Osirinsky and Kleitman published their findings in the journal Science. The paper was short, almost modest, but what it described was revolutionary. They had identified a distinct, previously unknown stage of sleep characterized by rapid eye movements, fast brain activity, and dreaming. They called it REM sleep, rapid eye movement sleep, and it turned the entire field on its head. Because up until that point, remember, sleep researchers had been mapping out these progressively deeper stages of non-REM sleep, going from light stage 1 down through the deep, synchronized, slow waves of stage 3 and 4. The assumption was that sleep was basically a one-way descent into deeper and deeper unconsciousness, followed by a gradual return to wakefulness, a U-shape, down and then up. But REM sleep didn't fit that model at all. It wasn't deeper than slow-wave sleep. In many ways, it seemed shallower, with brain activity that resembled waking. And yet it wasn't wakefulness either, because the person was clearly asleep, often more difficult to wake than during lighter non-REM stages. It was something else entirely, a third state of consciousness that was neither sleeping in the traditional sense nor awake, and it cycled. That's the other thing they discovered. REM sleep didn't just happen once during the night. It came and went in regular intervals, roughly every 90 minutes, alternating with periods of non-REM sleep in a repeating pattern that continued throughout the entire night. The first REM period might last only 10 minutes or so, but as the night went on, the REM periods got longer and the deep, slow-wave periods got shorter, so that by the early morning hours, you were spending most of your time in REM sleep. That's why your most vivid dreams tend to happen in the last few hours before you wake up. Think about what that means for a second. Your brain isn't just passively resting during sleep. It's cycling through this elaborate, precisely timed sequence of distinct neurological states, each with its own characteristic brain waves, its own pattern of eye movements, its own level of muscle tone. And that last part, muscle tone, turned out to be another key piece of the puzzle. During REM sleep, your body is essentially paralyzed. Not your diaphragm, you keep breathing, and not your eye muscles, obviously, but virtually every other voluntary muscle in your body goes limp. The technical term is atonia, and it's caused by active inhibition of motor neurons in your brainstem. Your brain is literally sending signals that prevent your muscles from moving. And here's what's really clever about that. Think about what would happen if you didn't have this paralysis. During REM sleep, your brain is generating vivid, immersive experiences. Dreams where you're running, climbing, fighting, flying. If your motor system were still connected, you'd be acting all of that out. You'd be thrashing around in bed, swinging your arms, kicking your legs, which, by the way, is exactly what happens in a condition called REM sleep behavior disorder, where this paralysis mechanism breaks down. It was formally described much later, in 1986, by Carlos Schenk and Mark Mahold at the University of Minnesota, and it's genuinely dangerous. People with this condition have injured themselves and their bed partners by physically acting out violent dreams. They'll punch walls, leap out of bed, knock over furniture, all while completely asleep. It's a vivid and somewhat unsettling demonstration of just how active the brain really is during REM sleep, and how important that protective paralysis is. But let's go back to the 1950s for a moment, because the discovery of REM sleep opened up a cascade of questions that researchers are still working on today. The most obvious one being, why? Why does the brain do this? Why does it cycle through these elaborate stages? What is each stage actually for? Now, Kleitman and Oserinsky had found the phenomenon, but explaining it was a different matter entirely. And the person who probably did the most to map out the architecture of sleep in those early years was a young researcher named William Dement, who started as a medical student working in Kleitman's lab and went on to become, arguably, the most important sleep researcher of the 20th century. Dement was the one who really systematized the stages of sleep, who spent night after night in the lab meticulously recording the brain waves of sleeping subjects and mapping out exactly how the stages cycled. He documented the full sleep cycle, the descent from wakefulness through stage 1, stage 2, into deep, slow-wave sleep, then back up through the lighter stages and into REM, then the whole thing repeating, roughly every 90 minutes, 4 or 5 times a night. He called this the sleep architecture, and it's a term we still use. Dement was also one of the first researchers to seriously study what happens when you take sleep away. Sleep deprivation experiments had been done before. There was a famous case in 1896 where 3 volunteers at the Iowa State College stayed awake for 90 hours while researchers measured their reaction times and cognitive performance. But Dement brought a new level of rigor to it. He selectively deprived people of specific sleep stages, particularly REM sleep, by waking them up every time the EEG showed they were entering a REM period, but letting them sleep normally otherwise. And what he found was striking. People who were deprived of REM sleep became increasingly desperate to get it. Each night, the brain would try to enter REM more and more frequently, and the researcher would have to wake the subject more and more often. On the first night, you might need to wake someone 5 or 6 times. By the 3rd or 4th night, you might be waking them 20 or 30 times, because the brain kept trying to plunge back into REM almost immediately after being woken, and when you finally let them sleep undisturbed, they'd have a massive REM rebound, spending far more time in REM than normal, as if the brain were catching up on something it desperately needed. This suggested that REM sleep wasn't optional. It wasn't some quirky byproduct of brain activity during sleep. It was something the brain actively needed and would fight to get. But needed for what? That question drove decades of research, and honestly, we still don't have a complete answer. But we have pieces, and some of them are fascinating. One of the most important clues came from studying who gets the most REM sleep, and the answer is babies. Newborn infants spend roughly 50% of their total sleep time in REM, compared to about 20 to 25% for adults. Premature infants spend even more, up to 80%, and this isn't just a human thing. Across mammalian species, the younger the animal, the more REM sleep it gets. A newborn kitten spends almost all of its sleep time in REM. This pattern led researchers to hypothesize that REM sleep plays a critical role in brain development, that all that neural activity during REM is somehow helping to wire up the developing brain, strengthening connections, pruning unnecessary ones, building the neural architecture that the animal will need for the rest of its life. And there's good evidence for this. During REM sleep, the brain is incredibly active, but it's active in a particular way. It's not just randomly firing. It's replaying and reorganizing patterns of activity, strengthening certain synaptic connections while weakening others. This process, which neuroscientists call synaptic plasticity, is fundamental to learning and memory. And this is where things start to connect to your everyday experience in a really direct way. Because it's not just developing brains that need this. Adult brains need it too. Here's a study that illustrates this beautifully. In the early 2000s, researchers at Harvard, led by Robert Stickgold, had subjects learn a visual texture discrimination task. Basically, you had to spot a subtle pattern on a screen, and your performance was measured by how quickly and accurately you could do it. Subjects practiced the task and then were tested again after a period of sleep or a period of wakefulness. And the results were clear. Subjects who slept between training and testing improved significantly. Subjects who stayed awake did not. But here's the really elegant part. The amount of improvement correlated specifically with the amount of time spent in certain sleep stages. Early night sleep, which is dominated by deep slow-wave sleep, and late night sleep, which is dominated by REM sleep. Both contributed to different aspects of the learning. It wasn't just one stage doing all the work. The different stages of sleep seemed to be handling different parts of the memory consolidation process. And this is where we start to see why that elaborate cycling matters, why the brain doesn't just drop into one state and stay there all night. Each stage seems to be doing something different. Deep, slow-wave sleep appears to be particularly important for consolidating declarative memories, facts, events, things you can consciously recall. REM sleep seems more involved in procedural memory, emotional processing, and creative problem-solving. And stage 2, non-REM sleep, with those distinctive sleep spindles that Berger's successors discovered, seems to play its own role in integrating new information with existing knowledge. So your brain isn't just resting during sleep, it's working. It's sorting through the experiences of the day, deciding what to keep and what to discard, strengthening the neural pathways that encode important memories and letting the unimportant ones fade. It's like a librarian working the night shift, filing away new books, cross-referencing them with existing collections, occasionally pulling something off the shelf to re-examine it in a new light. And this process requires the full symphony of sleep stages, playing out in their precise 90-minute cycles, each one contributing something essential. Which brings us to something that might seem like a completely different topic, but is actually deeply connected: the question of what controls the timing of sleep. Because it's not just that you get sleepy when you've been awake for a long time, although that's part of it. There's another system at work, one that operates on a roughly 24-hour cycle, ticking away inside your body like a clock. And in fact, that's exactly what scientists call it, the circadian clock. The word circadian comes from the Latin circadium, meaning about a day. And the story of how we discovered this internal timekeeper is one of the most elegant in all of biology. It starts, of all places, with a French astronomer in 1729, a man named Jean-Jacques d'Ortu de Méran, who noticed something peculiar about a mimosa plant sitting on his windowsill. The plant's leaves would open during the day and close at night, which wasn't surprising. Lots of plants do that. But de Méran wondered what would happen if he put the plant in complete darkness. Would the leaves stay closed? Or would they continue to open and close on their own, even without any light to guide them? And here's the beautiful thing: they did. The leaves kept right on opening and closing, roughly on schedule, as if the plant somehow knew what time it was, even sealed away in a dark cupboard where no sunlight could reach it. De Mairan presented his findings to the French Royal Academy of Sciences, and it was one of those observations that was so simple, so clean, that it should have changed everything. But it didn't. Not right away. Because the obvious objection was that maybe the plant was sensing something else. Temperature changes, vibrations, some subtle cue leaking through. Maybe it wasn't really an internal clock at all. And that objection lingered for a very long time. It took more than 2 centuries, actually, for the scientific world to fully grapple with what de Mairan's mimosa was telling us. In the 1960s, a German biologist named Jürgen Aschoff decided to do with humans what de Mairan had done with plants. He built an underground bunker in the Bavarian countryside, literally a reinforced concrete apartment beneath the earth, completely isolated from natural light, temperature fluctuations, and any external time cues. No clocks, no radio, no newspapers. And then he invited volunteers to live there, sometimes for weeks at a time, just going about their daily routines, eating, sleeping, reading, whatever they wanted, with no way of knowing whether it was day or night outside. What Aschoff found was remarkable. His subjects didn't just collapse into random patterns of sleeping and waking. They maintained a cycle. They'd sleep, wake up, stay active for a stretch, then sleep again, over and over, in a rhythm that was almost, but not quite, 24 hours. Most people drifted to something closer to 25 hours, though later, more carefully controlled studies revised that estimate down to about 24 hours and 11 minutes. The point is, even without any external signals, the body kept time. Not perfectly, but persistently. There was a clock inside, and it was running. Now you might be wondering, where is this clock? Like, physically? Where in the body does it live? And this is where the story gets wonderfully specific. In the early 1970s, 2 independent research groups, one led by Robert Moore and Victor Eichler, the other by Friedrich Steffen and Irving Zucker, were working with rats, and they zeroed in on a tiny cluster of neurons in the brain called the suprachiasmatic nucleus. The name sounds intimidating, but it just means above the optic chiasm, which is the spot where the optic nerves from your 2 eyes cross over each other on their way to the back of the brain. And sitting right above that crossing point, nestled in the hypothalamus, is this little bundle of about 20,000 neurons. 20,000. That's it. In a brain with roughly 86 billion neurons, this minuscule cluster acts as the master pacemaker for your entire body. When Moore and Eichler destroyed the suprachiasmatic nucleus in rats, you'll sometimes see it abbreviated as the SCN. The animals lost all sense of circadian rhythm. They'd sleep and wake in random fragments, eat at odd times. Their hormone cycles fell apart. The clock was gone. And here's what confirmed it beyond any doubt. In the 1990s, a researcher named Martin Ralph at the University of Virginia took SCN tissue from mutant hamsters that had an abnormally short circadian cycle, about 20 hours instead of 24, and transplanted it into hamsters whose own SCN had been destroyed. The recipient hamsters adopted the donor's 20-hour rhythm. The clock wasn't just necessary for circadian timing, it was sufficient. You could literally transplant someone else's clock and run on their schedule. So how does this clock actually work at the molecular level? And this is genuinely one of the most beautiful pieces of biology ever uncovered. Inside each of those 20,000 SCN neurons, there's a feedback loop made of proteins. The key players were identified through decades of painstaking genetics work, much of it done in fruit flies, because flies have circadian rhythms too. They're active during the day and rest at night, and you can breed millions of them and screen for mutations. In the early 1970s, Seymour Benzer and his student Ron Konopka at Caltech identified the first clock gene, which they simply called period. Flies with mutations in this gene either had no rhythm at all, or their rhythms were too short or too long. But it took until 1984 for Michael Rosbash and Jeffrey Hall at Brandeis, along with Michael Young at Rockefeller University, to clone the gene and start figuring out what it actually did. Here's the basic idea, and it's so elegant it almost feels like it was designed by an engineer. The period gene produces a protein called PER, and that protein gradually accumulates in the cell over the course of several hours. As PER builds up, it eventually enters the nucleus of the cell and shuts down the very gene that's making it. So the gene turns itself off through its own product. Then, with the gene silenced, no new PER protein is being made, and the existing PER slowly degrades. Once enough of it has broken down, the gene is no longer being suppressed, so it switches back on, and the whole cycle starts again. One full loop of production, accumulation, suppression, and degradation takes, you guessed it, about 24 hours. Think about that for a second. A single molecular feedback loop, ticking away inside individual cells, keeping time with roughly the same period as the rotation of the Earth. It's like a tiny biochemical pendulum. And it's not just in your brain. Virtually every cell in your body has its own version of this clock. Your liver cells have it. Your skin cells have it. Your heart cells have it. The SCN acts as the conductor, synchronizing all these peripheral clocks, but the clockwork itself is everywhere. Rosbash, Hall, and Young won the Nobel Prize in Physiology or Medicine in 2017 for this work, and honestly, it was long overdue. They'd spent decades unraveling the mechanism, discovering additional components, proteins called TIM, CRY, CLOCK, and BMAL1 that add layers of regulation and fine-tuning to that core feedback loop. It's not just one simple loop, it's interlocking loops with checks and balances, temperature compensation so the clock doesn't speed up when you have a fever, and input pathways that allow external signals to adjust the timing. And the most important of those external signals is, of course, light. This brings us back to the anatomy. Remember how the SCN sits right above the optic chiasm? That's not a coincidence. There's a dedicated neural pathway running from special cells in your retina directly to the SCN. And these aren't the rods and cones you learned about in school, the ones responsible for vision. These are a completely different type of photoreceptor, discovered only in the early 2000s, called intrinsically photosensitive retinal ganglion cells. They contain a pigment called melanopsin, and they're not interested in forming images. They don't care about shapes or colors or faces. They're measuring one thing: the overall brightness of the ambient light, particularly light in the blue part of the spectrum, around 480 nanometers wavelength. When blue-enriched light hits these melanopsin cells, they send a signal to the SCN that essentially says, "It's daytime." And the SCN uses this signal to reset the clock each day, nudging it forward or backward so that your internal rhythm stays locked to the actual 24-hour light-dark cycle of the planet. This process is called entrainment, and it's the reason you can adjust to jet lag. Your clock gets reset a little bit each day until it matches local time. It typically takes about 1 day per time zone crossed, which is why flying from New York to London feels rough for about 5 days, but eventually resolves. And here's where this connects back to something deeply practical, something that affects you every single night. When the SCN senses that light levels are dropping, that evening is arriving, it sends a signal to a small gland deep in the center of your brain called the pineal gland. René Descartes, back in the 17th century, thought the pineal gland was the seat of the soul, which turned out to be wrong in the most spectacular way, but the gland does have an important job. When the SCN gives the go-ahead, the pineal gland begins secreting melatonin, a hormone that seeps into your bloodstream and tells the rest of your body that night has arrived. Melatonin doesn't knock you out. That's a common misconception. It's more like a signal, a chemical announcement that says, the conditions are right for sleep now. Start winding down. This is why staring at your phone in bed is such a problem, and I say that with zero judgment because everyone does it. That screen is pumping blue-enriched light directly into your melanopsin cells, and those cells are telling your SCN that it's still daytime, and your SCN is telling your pineal gland to hold off on the melatonin. You're essentially lying to your own circadian system. The architecture of sleep, all those carefully orchestrated stages we talked about, the slow waves, the spindles, the REM periods, it all depends on proper circadian timing. When you shift that timing, you don't just fall asleep later, you change the structure of the sleep itself. But the circadian clock is only half the story of sleep timing. There's a second system running in parallel, and understanding how these two systems interact is one of the most important insights in all of sleep science. It was formalized by a Hungarian-born sleep researcher named Alexander Borbély, working at the University of Zurich in a landmark 1982 paper that proposed what he called the two-process model of sleep regulation. And the beauty of Borbély's model is its simplicity. He said there are exactly two forces that determine when you sleep and when you wake. Process C is the circadian rhythm we've just been talking about. That 24-hour oscillation driven by the SCN. Process S is something different. Process S is sleep pressure, and it's governed by a molecule that had been hiding in plain sight for decades, one that every single one of us produces every single day just by being awake. Adenosine. That's the molecule. And the story of how we figured out what adenosine does is one of those wonderful cases where the answer was sitting right there in our coffee cups the whole time. So let's talk about adenosine for a minute, because it's genuinely fascinating. Adenosine is a nucleoside. It's one of the building blocks of ATP, adenosine triphosphate, which is the primary energy currency of every cell in your body. When your neurons fire, when your brain does its work of thinking and perceiving and remembering and just keeping you alive, it burns through ATP. And as ATP gets broken down, adenosine accumulates as a byproduct. Think of it like ash building up in a fireplace. The longer the fire burns, the more ash there is. The longer you're awake, the more adenosine builds up in your brain, particularly in a region called the basal forebrain. And adenosine doesn't just sit there passively. It binds to specific receptors, adenosine receptors, especially the A1 and A2A subtypes, on the surface of neurons that are responsible for keeping you alert. When adenosine latches onto those receptors, it inhibits those wake-promoting neurons. It's essentially a chemical record of how long you've been awake, and the longer that record grows, the stronger the signal becomes. You need to sleep. That heaviness you feel behind your eyes after a long day, that fog that rolls in when you've been up for 16 or 17 hours, that's adenosine talking. And here's where the coffee comes in. Caffeine, the most widely consumed psychoactive drug on the planet, works because its molecular structure is remarkably similar to adenosine. similar enough that caffeine can slide into those same A1 and A2A receptors, like a key that fits the lock but doesn't turn it. Caffeine blocks the receptor without activating it. So the adenosine is still there, still accumulating, but your brain can't feel it. It's like putting earplugs in while the fire alarm is going off. The alarm hasn't stopped. You just can't hear it. This is why the caffeine crash is a real thing. When the caffeine eventually gets metabolized and clears out of those receptors, all that built-up adenosine floods in at once, and the sleepiness hits you like a wall. You didn't eliminate the sleep pressure. You just postponed your awareness of it. Now, Borbély's insight was to take this idea, this accumulating sleep pressure, process S, and map it against the circadian rhythm, process C, and show that sleep and wakefulness emerge from the interaction between these two forces. Here's how it works. Imagine two curves on a graph, though since your eyes might be closed, let me just describe what's happening. Process S, sleep pressure, rises steadily the longer you're awake. It starts low when you wake up in the morning and climbs throughout the day. Process C, the circadian alerting signal, also fluctuates, but on its own 24-hour rhythm. During the day, your SCN is actively promoting wakefulness. It's pushing against the rising tide of adenosine, keeping you alert even as sleep pressure builds. This is why you don't get progressively sleepier in a perfectly linear way throughout the day. You might actually feel more alert at 4 in the afternoon than you did at 11 in the morning, even though you've been awake longer, because the circadian alerting signal is peaking in the late afternoon and early evening. Some people call this the wake maintenance zone or the forbidden zone for sleep. It's the period in the evening, usually a couple of hours before your habitual bedtime, when it's actually hardest to fall asleep, even though your adenosine levels are near their daily peak. Your circadian system is pushing back that hard. But then, as evening progresses and melatonin starts to rise, and the circadian alerting signal begins to drop, the two curves cross. Sleep pressure is high, circadian alerting is falling away, and that convergence is what tips you into sleep. It's elegant, really. Two independent systems, running on completely different mechanisms. One tracking time of day through light and molecular clocks, the other tracking time awake through metabolic byproducts. And together they create a stable, predictable pattern of sleeping and waking. Borbély showed that you could predict sleep timing and sleep intensity remarkably well just from these two variables. And here's what's really clever about the model. It also explains what happens when things go wrong. Think about jet lag. When you fly across 6 time zones, your circadian clock is still set to your departure city. Process C is telling your brain it's afternoon, but the local clock says it's 10 at night, and your adenosine levels are through the roof because you've been traveling all day. The two processes are completely misaligned. That's why jet lag feels so uniquely awful. It's not just tiredness. It's a fundamental disagreement between two systems that are supposed to work in concert, and it takes days to resolve because the SCN can only shift about 1 to 1.5 hours per day in response to the new light-dark cycle. So a 6-hour time zone change takes roughly 4 to 6 days to fully adjust to. And during that entire period, your sleep architecture is disrupted. You're getting sleep, but the stages aren't properly organized because the circadian timing is off. Shift work is even worse, and this is where the conversation gets serious. Millions of people, nurses, factory workers, pilots, truck drivers, first responders, work schedules that put them at war with their own biology. A night shift worker is trying to sleep during the day, when their circadian system is aggressively promoting wakefulness, and trying to stay alert at night, when their circadian system is pulling them towards sleep. The adenosine system doesn't care what shift you work. It just tracks how long you've been awake. But the circadian system is anchored to the light-dark cycle, and unless you can completely control your light exposure, blackout curtains, timed bright light, avoiding sunlight on the commute home, the SCN never fully adjusts to a night shift schedule. Studies have shown that even long-term night shift workers, people who've been on the schedule for years, often have circadian rhythms that are only partially shifted. They're living in a state of chronic misalignment, and the consequences are not trivial. The World Health Organization classified night shift work as a probable carcinogen in 2007, based on evidence linking chronic circadian disruption to increased rates of breast cancer, colorectal cancer, and cardiovascular disease. There's elevated risk of metabolic syndrome, type 2 diabetes, depression, and impaired immune function. Some of this is mediated through sleep loss itself, and some through the disruption of circadian regulated processes that have nothing to do with sleep, things like DNA repair, cell division timing, and immune surveillance, all of which are under circadian control. The body doesn't just use the clock for sleep. It uses the clock for everything, and when you break the clock, everything downstream suffers. This brings us back to something I mentioned earlier, that 19th century idea about sleep being related to the clearance of toxins. Because it turns out those early researchers were onto something, even if they didn't have the tools to prove it. In 2012, a neuroscientist named Maiken Nedergaard, working at the University of Rochester, discovered something remarkable. She and her team identified a waste clearance system in the brain that they called the glymphatic system, a play on glial and lymphatic, because it's a network of channels formed by glial cells, specifically astrocytes, that allows cerebrospinal fluid to flow through brain tissue and flush out metabolic waste products. And here's the key finding. The glymphatic system is dramatically more active during sleep than during wakefulness. Nedergaard's team showed, initially in mice, that during sleep, the interstitial spaces in the brain, the gaps between cells, expand by as much as 60%, allowing cerebrospinal fluid to flow much more freely and carry away waste products that accumulate during waking hours. And one of the waste products that the glymphatic system clears is beta amyloid. If that name sounds familiar, it should. Beta amyloid plaques are one of the hallmark pathological features of Alzheimer's disease. They're sticky protein fragments that accumulate between neurons and are associated with cognitive decline and neurodegeneration. Nedergaard's work suggested a provocative connection. If the brain clears beta amyloid during sleep, and if chronic sleep disruption impairs that clearance, could poor sleep contribute to the development of Alzheimer's? The research is still ongoing, but the epidemiological evidence is striking. Multiple large studies have found that people who consistently sleep fewer than 6 hours per night in midlife have significantly elevated risk of developing dementia later in life. Matthew Walker and others have shown that even a single night of sleep deprivation increases beta amyloid levels in the human brain, as measured by PET imaging. The relationship appears to be bidirectional. Alzheimer's pathology itself disrupts sleep, which may further impair clearance, creating a vicious cycle. Think about what that means for a moment. Sleep isn't just rest. It isn't just memory consolidation or emotional regulation or physical recovery, though it's all of those things. It's also, quite literally, the time when your brain takes out the trash. Those 19th century researchers who thought sleep had something to do with clearing accumulated substances from the brain. They were right. They just didn't know the mechanism, and they didn't have the imaging technology to watch it happen. It took until 2012, almost 130 years after those early toxin theories, for the glymphatic system to be identified and for that intuition to be vindicated. And Nedergaard's discovery opened up an entirely new way of thinking about why we sleep. Because the question of why we sleep, really fundamentally, why evolution would design an organism to spend a third of its life unconscious and vulnerable has haunted sleep science from the beginning. Alan Rechtschaffen, one of the pioneers of sleep research at the University of Chicago, famously said, "If sleep does not serve an absolutely vital function, then it is the biggest mistake the evolutionary process has ever made." Every animal that has been studied sleeps or exhibits something functionally equivalent to sleep. Dolphins sleep with one hemisphere at a time so they can keep swimming and breathing. Migrating birds can sleep during flight, in microsecond bursts. Fruit flies sleep. Jellyfish exhibit sleep-like states. Whatever sleep does, it's so important that evolution has never found a way to eliminate it, even in species where being unconscious is extraordinarily dangerous. And the answer increasingly seems to be that sleep doesn't serve one function, it serves many. The glymphatic clearance is one. Memory consolidation is another. Emotional processing, synaptic homeostasis, immune regulation, hormonal balance, tissue repair. The list keeps growing. Giulio Tononi and Chiara Sorelli at the University of Wisconsin proposed something called the synaptic homeostasis hypothesis, which offers yet another piece of the puzzle. And it's one of those ideas that's so elegantly simple that once you hear it, you kind of can't unhear it. It reframes the whole question of what sleep is for. So here's the basic idea. During the day, when you're awake, your brain is learning. Constantly. Every conversation, every glance out the window, every problem you solve, every new face you see, all of that learning involves strengthening synaptic connections between neurons. That's what learning is, at the cellular level. When two neurons fire together repeatedly, the connection between them gets stronger. The synapse gets more efficient, more responsive. You've probably heard some version of the phrase, neurons that fire together wire together. And that's a simplification, but it captures the essence. Throughout the waking day, synapses across your brain are being potentiated, strengthened, upregulated, made louder, essentially. Now here's the problem. Strengthening synapses costs energy. Bigger synapses require more neurotransmitter, more receptor proteins, more cellular machinery to maintain. And if all you ever did was strengthen connections, if every day just added more and more synaptic weight, you'd run into trouble pretty quickly. The brain would become metabolically unsustainable. It would be like turning up the volume on every channel at once. Eventually, everything is loud and nothing stands out. The signal-to-noise ratio collapses. You can't distinguish important patterns from background noise because everything has been amplified equally. What Tononi and Sorelli proposed, starting around 2003 and refining the idea over the next 2 decades, is that sleep, particularly deep, slow-wave sleep, serves as a kind of synaptic reset. During those big rolling delta waves, the brain systematically scales down synaptic strength across the board. Not erasing anything, not wiping the slate clean, but turning the overall volume back down to a sustainable baseline. Think of it like this. During the day, you highlight passages in a book. By evening, you've highlighted so much that the highlighting has lost its meaning. During sleep, the highlighting fades. But the passages you highlighted most intensely, the ones that were reinforced multiple times or tagged as emotionally important, those retain more of their color. The relative differences are preserved, but the absolute level comes back down. So you wake up the next morning with a brain that's ready to learn again, that has capacity for new synaptic strengthening, and that has kept the most important patterns from the day before while letting the trivial ones fade. It's a beautiful idea. And what makes it more than just a nice theory is that Tononi and Cirelli have gathered substantial evidence for it. They've shown, in both animal models and human studies, that synaptic strength, measured by things like the size of dendritic spines, the density of certain receptor proteins, and the amplitude of neural responses, is higher after a period of wakefulness and lower after a period of sleep. In fruit flies, they've literally measured the physical size of synapses and found they grow during waking hours and shrink during sleep. In mice, electron microscopy studies from their lab showed that the average synapse is roughly 18% smaller after sleep than after an equivalent period of wakefulness. 18%. That's not a subtle effect. And here's what ties it back to those slow waves we talked about earlier. The slow oscillations of deep sleep, those synchronized pulses where huge populations of neurons alternate between firing together and going silent, appear to be the mechanism by which this downscaling happens. The rhythmic on-off cycling is thought to preferentially weaken the synapses that are weakest, the ones that were only mildly potentiated during the day, while relatively sparing the ones that were strongly reinforced. It's like the slow waves are running a sorting algorithm, and the criterion is, how important was this connection? How many times was it activated? Was it associated with reward, or emotion, or repeated practice? If so, it survives the night mostly intact. If it was just noise, a random correlation, an incidental perception, it gets pruned back. Now, this connects to memory consolidation in a really interesting way. You might remember that we talked about how slow-wave sleep seems especially important for declarative memory, facts, events, things you can consciously recall. The synaptic homeostasis hypothesis doesn't contradict that. It actually enriches it, because one way to consolidate a memory is to make sure the important synaptic patterns are preserved while the unimportant ones are cleared away. Consolidation isn't just about strengthening the right connections. It's about weakening the wrong ones. It's about improving contrast. And that's exactly what synaptic downscaling would accomplish. Tononi, by the way, is a fascinating figure in his own right. He's an Italian-born neuroscientist who trained with the Nobel laureate Gerald Edelman before moving to the University of Wisconsin-Madison. He's also the originator of integrated information theory, which is one of the leading scientific theories of consciousness, a framework for understanding what consciousness actually is, mathematically. So when Tononi thinks about sleep, he's thinking about it in the context of this much larger question. What does it mean for a brain to be conscious? And what changes when consciousness fades during deep sleep? His approach to sleep science is deeply theoretical, almost philosophical, but grounded in hard data. Cirelli, his longtime collaborator and partner, is equally rigorous, a physician-scientist who brings the molecular biology expertise that gives their hypotheses real mechanistic teeth. And this idea of synaptic homeostasis, this notion that sleep is fundamentally about restoring the brain's capacity to learn, it gives you yet another angle on why sleep deprivation is so devastating. If you don't sleep, you don't downscale. Your synapses stay saturated. Your brain literally runs out of room to encode new information efficiently. And sure enough, one of the most consistent findings in sleep deprivation research is that learning ability tanks. Not just because you're tired and can't pay attention, although that's part of it, but because the synaptic substrate for learning has been compromised. The hardware is maxed out. So let's step back for a moment and appreciate where we've arrived. We started with the ancient Greeks thinking sleep was a passive state, a little death, something that just happened when the body ran down. And now we're looking at sleep as this incredibly active, multi-layered process. The glymphatic system is flushing metabolic waste. Memory traces are being replayed and consolidated across different stages. Synapses are being selectively downscaled to maintain the brain's learning capacity. Emotional memories are being reprocessed, stripped of their raw affective charge while the informational content is preserved. And all of this is happening in an orchestrated sequence, timed to the architecture of sleep cycles, the 90-minute rhythm of NREM and REM that Dement mapped out decades ago. But there's another dimension to sleep's function that we haven't really explored yet, and it's one that affects your body just as profoundly as everything we've discussed affects your brain. Because sleep isn't just a brain phenomenon, it's a whole body phenomenon. And some of the most consequential health discoveries of the last 20 years have come from understanding what happens to the rest of your body when you sleep, and what goes wrong when you don't. Let's talk about hormones for a moment. Growth hormone, for instance. In adults, the largest pulse of growth hormone secretion occurs during the first bout of deep, slow-wave sleep, typically within the first hour or so after you fall asleep. This isn't a coincidence. Growth hormone is essential for tissue repair, muscle recovery, bone maintenance, and cellular regeneration. If you're an athlete, or if you've had surgery, or if you're a child whose body is literally growing, that first deep sleep pulse of growth hormone is doing critical work. Disrupt someone's slow-wave sleep, even if you let them sleep the same total number of hours, and you blunt that growth hormone pulse significantly. Eve Van Couter's lab at the University of Chicago demonstrated this beautifully in a series of studies in the late 1990s and 2000s. She showed that when you restrict young, healthy adults to 4 hours of sleep per night for just 6 nights, their glucose tolerance deteriorates to the point where they look pre-diabetic. Their insulin sensitivity drops. Their cortisol levels in the evening, which should be low because cortisol follows its own circadian rhythm, peaking in the morning and falling at night, those evening cortisol levels stay elevated. And elevated evening cortisol is associated with insulin resistance, immune suppression, and accelerated aging. Van Couter's work was genuinely alarming, and it helped catalyze a whole field of research into the metabolic consequences of insufficient sleep. Here's one finding that really sticks with me. When you're sleep deprived, your levels of leptin, the hormone that signals satiety, that tells your brain you've eaten enough, those levels drop. And your levels of ghrelin, the hormone that signals hunger, those levels rise. So sleep deprivation doesn't just make you tired, it makes you hungry. Specifically, it makes you crave high-calorie, high-carbohydrate foods. Your brain's reward centers become more responsive to food cues. Your prefrontal cortex, which is supposed to exercise judgment and restraint, is already impaired from the sleep loss. It's a perfect storm for overeating. And the epidemiological data bears this out. Short sleep duration is one of the strongest risk factors for obesity, independent of diet and exercise. Think about what that means from an evolutionary perspective. For most of human history, food scarcity was the norm. If you were awake longer, if something was keeping you from sleeping, like a threat or a migration or a famine. It would make biological sense for your body to increase hunger signals and drive you to seek calorie-dense food. The hormonal response to sleep deprivation is probably an ancient survival mechanism, but in a modern environment where high-calorie food is available 24 hours a day, and the thing keeping you awake is your phone or your work schedule, that same mechanism becomes pathological. And it's not just metabolism. The immune system is profoundly affected by sleep. If you've ever noticed that you're more likely to catch a cold when you've been burning the candle at both ends, that's not your imagination. Sheldon Cohen at Carnegie Mellon ran a remarkable study where he actually exposed volunteers to rhinovirus, the common cold virus, after monitoring their sleep for 2 weeks. The people who had been sleeping less than 7 hours a night were nearly 3 times more likely to develop a cold than those sleeping 8 hours or more. 3 times. And this was after controlling for other variables like stress, age, smoking, and baseline antibody levels. Sleep was the dominant factor. Your immune system does some of its most important work while you sleep. Certain cytokines, signaling molecules that coordinate immune responses, are produced and released during sleep. T-cell function improves. The body's inflammatory response is modulated. There's even evidence that sleep enhances the effectiveness of vaccines. A study by Arik Prather and colleagues showed that people who slept fewer than 6 hours the night after receiving a hepatitis B vaccine produced less than half the antibody response compared to those who slept normally, and that diminished response was still detectable months later. So if you've ever gotten a flu shot and wondered whether it actually worked, part of the answer might depend on how well you slept that week. All of this, the metabolic effects, the immune effects, the hormonal cascades, it all reinforces this picture of sleep as a whole organism maintenance process. Your brain is consolidating memories and clearing waste and downscaling synapses, yes. But simultaneously, your body is repairing tissue, calibrating hunger signals, replenishing immune defenses, regulating inflammation, and resetting hormonal rhythms. It's all happening in parallel, choreographed by the same circadian and homeostatic systems we've been tracing throughout this story. And this brings us to something that Matthew Walker, the neuroscientist at UC Berkeley, has been particularly vocal about, which is the question of what happens when a society systematically undervalues sleep. Because that's what modern industrial societies have done. Thomas Edison reportedly bragged about sleeping only 4 hours a night and called sleep a waste of time. Margaret Thatcher famously claimed she needed only 4 hours. There's a persistent cultural mythology that successful people sleep less, that sleep is for the weak, that you can sleep when you're dead. And Walker's argument, backed by an enormous body of evidence, is that this attitude is literally killing people. Cardiovascular disease, cancer, Alzheimer's, diabetes, obesity, depression, anxiety. The list of conditions linked to chronic sleep insufficiency is staggering, and the mechanisms we've been discussing explain why. It's not mysterious. If you deprive the brain of its waste clearance window, amyloid accumulates. If you deprive the body of its hormonal reset, metabolic dysfunction follows. If you suppress immune function night after night, vulnerability to disease increases. But here's something that often gets lost in the conversation about sleep and health, and it has to do with the architecture of sleep itself, not just the quantity, but the quality. Because it turns out that not all sleep disruption is created equal, and some of the most insidious forms of sleep disruption don't reduce your total sleep time at all. They fragment it. They shatter the architecture without necessarily shortening the night. and the most common culprit, affecting an estimated 1 billion people worldwide, is something that wasn't even recognized as a medical condition until the 1960s and 70s. And even then, it took decades before the medical community truly understood how devastating it could be. We're talking about obstructive sleep apnea, and the story of how we came to understand sleep apnea is itself a fascinating window into how medicine sometimes takes an embarrassingly long time to see what's right in front of it. Because people have been snoring, loudly, dramatically, gasp and choke snoring, for as long as people have been sleeping. Dickens described it. He created a character in the Pickwick Papers, published in 1836, a boy named Joe, who was enormously fat, constantly drowsy, and snored like thunder. Joe would fall asleep in the middle of conversations, in the middle of meals, standing up. Dickens wrote him as comic relief, but what he was actually describing, with remarkable clinical accuracy, was a person suffering from severe obstructive sleep apnea. And for over a century after Dickens, the medical establishment basically treated people like Joe the same way, as oddities, as curiosities, sometimes as jokes. The fat man who snores, the husband whose wife sleeps in the other room. Nobody thought of it as a disease. That started to change in 1965, when a group of researchers in Europe, notably a French neurophysiologist named Christian Guilleminot and a team in Germany, began systematically studying patients who had excessive daytime sleepiness and loud snoring. They monitored these patients overnight with the tools that Oserinsky and Kleitman and Dement had developed, EEG, EOG, EMG, plus something new. They monitored breathing, and what they found was alarming. These patients would fall asleep, their muscles would relax, and then their upper airway would literally collapse. The soft tissue at the back of the throat, the tongue, the soft palate, the pharyngeal walls would sag inward under gravity and seal off the airway. The person would stop breathing. Not for a second or two, for 10 seconds, 20 seconds, sometimes 30 or 40 seconds. Their blood oxygen would drop, their heart rate would spike, and then their brain, sensing the emergency, would trigger a micro-arousal, a brief, partial awakening, just enough to restore muscle tone, reopen the airway, and gasp in a breath. The person might not even know they woke up. They'd make a snorting, choking sound, take a few breaths, fall back to sleep, and the whole cycle would repeat. 5 times an hour, 10 times an hour. In severe cases, 60, 70, 80 times an hour. All night long. Think about what that means for the architecture of sleep. Remember those beautiful 90-minute cycles we talked about? The descent through stage 1, stage 2, into deep, slow-wave sleep, then back up into REM, then down again. Each cycle building on the last, each stage performing its specific function. Memory consolidation, synaptic downscaling, waste clearance, hormonal release. Now imagine that every few minutes, a tiny alarm goes off in the brainstem. Not loud enough to fully wake you, not memorable enough to register in consciousness, but enough to yank you out of whatever stage you're in and reset the cycle. You never reach deep, slow-wave sleep, or you reach it only briefly before being pulled out. You barely touch REM. Your brain spends the entire night in the shallow waters of stage 1 and stage 2, never diving deep. And you wake up in the morning having been asleep for 8 hours, and you feel like you haven't slept at all. Because in a very real sense, you haven't. The quantity was there. The architecture was destroyed. Guilleminot, who eventually moved to Stanford and became one of the founding figures of sleep medicine, was among the first to connect these dots systematically. He coined the term obstructive sleep apnea syndrome and began publishing case studies in the early 1970s. And here's what's remarkable. The patients he described weren't just tired. They were cognitively impaired. They had trouble concentrating, trouble with memory, trouble with mood regulation. They had high blood pressure. They had heart arrhythmias. Some of them had been misdiagnosed with depression, with laziness, with personality disorders. Some had lost jobs because they kept falling asleep at work. Some had caused car accidents because they fell asleep at the wheel. And all of them had this same pattern on overnight monitoring: repeated airway collapse, repeated oxygen desaturation, repeated microarousals, and devastated sleep architecture. Now, the treatment options in the early days were pretty grim. If you had severe obstructive sleep apnea in the 1970s, your doctor might recommend a tracheostomy, literally cutting a hole in your throat below the obstruction so air could bypass the collapsing upper airway. It worked. Patients who got tracheostomies suddenly slept through the night. Their daytime sleepiness vanished. Their blood pressure improved. But it was a drastic, disfiguring surgery, and most patients, understandably, didn't want a permanent hole in their neck. So the condition remained largely untreatable for the majority of people who had it. And then, in 1981, a physician in Sydney, Australia, named Colin Sullivan, had an idea that was so simple, it almost seems obvious in retrospect, except nobody had thought of it before. Sullivan was studying obstructive sleep apnea in dogs. Yes, bulldogs, who are prone to it for the same anatomical reasons some humans are: their short snouts and crowded airways. And he realized that if you could deliver a continuous stream of pressurized air through the nose, you could essentially create a pneumatic splint. The air pressure would hold the soft tissue open from the inside, preventing the collapse. You wouldn't need surgery. You just need a mask and a blower. Sullivan built the first prototype in his lab. It was crude: a motor from a vacuum cleaner, some tubing, a mask fashioned from fiberglass resin molded to the patient's face. He tried it on a patient with severe sleep apnea who was scheduled for a tracheostomy. The patient put on this contraption, fell asleep, and for the first time in who knows how long, his airway stayed open, his oxygen stayed up, his sleep architecture normalized. He woke up feeling rested. Sullivan published the results in The Lancet in 1981, and CPAP, continuous positive airway pressure, was born. It's one of those moments in medical history that's genuinely beautiful in its simplicity. You have this devastating condition that fragments sleep, destroys quality of life, and increases the risk of heart attack, stroke, hypertension, diabetes, cognitive decline. And the solution is, essentially, a gentle fan blowing air through your nose. No drugs, no surgery, just physics. Pressure equals force over area. Keep the pressure above the critical closing pressure of the airway, and the tissue stays open. That's it. Of course, the early CPAP machines were loud, uncomfortable, and the masks were terrible. Patient compliance was a huge problem. Still is, honestly. A lot of people get prescribed CPAP, use it for a few nights, hate the sensation of sleeping with a mask strapped to their face, and shove it in a closet. But the technology has improved enormously since Sullivan's vacuum cleaner prototype. Modern machines are whisper quiet, auto-adjusting. They sense the pressure needed breath by breath and modulate accordingly. And the masks come in dozens of designs. And the evidence for what happens when apnea is properly treated is overwhelming. Blood pressure drops. Cardiovascular risk decreases. Daytime alertness returns. cognitive function improves. And here's a detail that connects back to everything we've been talking about. Studies have shown that effective CPAP treatment restores slow-wave sleep and REM sleep to near normal proportions. The architecture comes back. The cycles reassemble. The brain can finally do its nightly maintenance. But here's what's sobering. That estimate of 1 billion people worldwide with obstructive sleep apnea, that comes from a landmark 2019 study published in The Lancet Respiratory Medicine. 1 billion. And the vast majority of them are undiagnosed. They don't know they have it. They just know they're tired. They just know they can't concentrate. They just know their partner complains about their snoring. In the United States alone, estimates suggest that roughly 80% of moderate to severe cases are undiagnosed. 80%. These are people walking around with fragmented sleep architecture, elevated cortisol, suppressed growth hormone, impaired glymphatic clearance, disrupted memory consolidation, all the downstream consequences we've been cataloging, and they don't know why they feel the way they feel. And it's not just about weight, though obesity is a major risk factor because excess tissue around the neck narrows the airway. Anatomy matters. People with naturally narrow airways, recessed jaws, large tonsils, or particular craniofacial structures are at higher risk regardless of weight. Age matters. Muscle tone decreases as you get older, making collapse more likely. Men are at higher risk than women, though the gap narrows significantly after menopause, which tells you something about the protective role of hormones in maintaining airway muscle tone. Even children can have it, usually because of enlarged tonsils and adenoids, and in kids, the consequences can be particularly stark. Behavioral problems, attention deficits, poor academic performance, sometimes misdiagnosed as ADHD. Remove the tonsils, restore normal sleep architecture, and the behavioral problems often resolve. It's remarkable. So sleep apnea is this massive, largely hidden public health problem that illustrates a broader point Walker and others have been making. We don't just need more sleep. We need intact sleep. Unbroken sleep. Sleep with its architecture preserved. And this connects to something else that disrupts sleep architecture in ways we're only beginning to fully appreciate, which is the role of alcohol. Because a lot of people use alcohol as a sleep aid, a nightcap, a glass of wine to help them drift off. And it does help you fall asleep faster. Alcohol is a sedative. It enhances GABA activity, the brain's main inhibitory neurotransmitter, and it suppresses neural firing. You feel relaxed, drowsy, and you fall asleep quickly. But what alcohol does to sleep architecture is quietly devastating. It suppresses REM sleep, particularly in the first half of the night. It fragments sleep in the second half as the body metabolizes the alcohol and experiences a rebound in sympathetic nervous system activity. And it relaxes the muscles of the upper airway, which, and you can probably see where this is going, worsens obstructive sleep apnea in people who already have it and can actually induce apnea-like events in people who normally don't. So the nightcap that's supposed to help you sleep is actually dismantling the very architecture that makes sleep restorative. And this brings us to a question that researchers have been grappling with increasingly over the past couple of decades, which is the relationship between sleep and mental health. Specifically, the question of whether disrupted sleep is a symptom of psychiatric illness or a cause of it. Because for a very long time, the clinical assumption was straightforward. Depression causes insomnia, anxiety causes insomnia, PTSD causes nightmares. Fix the psychiatric condition, and the sleep problems will resolve. Sleep disruption was downstream. It was a symptom, not a driver. But the evidence that's accumulated since the early 2000s has started to seriously challenge that assumption. And the shift is profound. It doesn't just change how we think about sleep, it changes how we think about mental illness itself. One of the key figures in this reorientation is Matthew Walker, who we've already encountered, but also Daniel Freeman, a clinical psychologist at the University of Oxford. Freeman and his colleagues published a landmark study in The Lancet Psychiatry in 2017, a randomized controlled trial involving over 3,600 university students with insomnia. They used cognitive behavioral therapy for insomnia, which is usually abbreviated CBTI, and it's essentially a structured program that helps people rebuild healthy sleep habits without medication. No sleeping pills, just behavioral and cognitive techniques. Things like stimulus control, sleep restriction, cognitive restructuring of anxious thoughts about sleep. And what Freeman found was striking. Treating the insomnia didn't just improve sleep. It significantly reduced symptoms of paranoia, hallucinations, anxiety, depression, and nightmares. The effect sizes were large. Improving sleep led to measurable improvements across a whole spectrum of mental health outcomes. And crucially, the causal direction was clear in this study because they intervened on sleep first and watched the psychiatric symptoms respond. Now, think about what that means. For decades, clinicians treated insomnia as a secondary complaint. You'd go to your doctor and say, "I can't sleep," and they'd ask about your mood, your stress, your life circumstances. And if you were depressed, the insomnia got filed under symptoms of depression. The treatment targeted the depression, usually with SSRIs or therapy, and the implicit assumption was that once the depression lifted, the sleep would come back. Sometimes it did, but often it didn't. And in many cases, the lingering insomnia was actually the thing that predicted relapse. People whose sleep didn't normalize after depression, treatment were far more likely to become depressed again. This pattern showed up over and over in longitudinal studies. A meta-analysis published in 2011 by Baglioni and colleagues, looking at data from over 20 studies and more than 100,000 participants, found that people with insomnia who were not currently depressed had a twofold increased risk of developing depression in the future. Insomnia wasn't just a symptom, it was a risk factor. It preceded the depression, and that's a fundamentally different clinical picture. Here's the thing. When you understand what sleep does at the neural level, this actually makes a lot of sense. Remember the role of REM sleep in emotional memory processing? Robert Stickgold's work, and also the work of Els van der Helm and Matthew Walker at Berkeley, showed that REM sleep helps strip the emotional charge from difficult memories. You experience something painful during the day, and during REM sleep that night, the brain replays the memory, but in a neurochemical environment where no norepinephrine, the brain's version of adrenaline, is essentially turned off. The amygdala is active, the memory is being processed, but without the stress chemistry. So the memory gets consolidated, but the sharp emotional edge gets softened. You wake up and the memory is still there, but it doesn't sting quite as much. Walker has called REM sleep overnight therapy, which is a lovely phrase, and it captures something real about the function. Now imagine what happens when that process is disrupted. If you're not getting enough REM sleep, because of alcohol, because of sleep apnea, because of chronic short sleep, because of medications that suppress REM, those emotional memories don't get properly processed. The emotional charge doesn't fade. You wake up and yesterday's pain is still just as raw, and the next day's pain stacks on top of it, and the day after that. you start to see how a person could spiral. The world starts to feel more threatening, more overwhelming, more emotionally saturated, because the neural machinery that's supposed to be recalibrating your emotional responses every night isn't getting the chance to do its job. Vanderhelm and Walker published a study in 2010 showing that sleep-deprived participants had a roughly 60% greater amygdala response to negative emotional images compared to well-rested participants. 60%. And the prefrontal cortex, the part of the brain that normally exerts top-down control over the amygdala, that helps you regulate your emotional reactions, that lets you pause and think before reacting. Its connectivity with the amygdala was significantly weakened after sleep deprivation. So you get a double hit. The emotional centers of the brain become hyperreactive, and the rational regulatory centers lose their ability to modulate that reactivity. It's like removing the brakes and pressing the accelerator at the same time, and this connects directly to anxiety. If you've ever had a night of terrible sleep and noticed that the next day everything felt slightly more alarming, you were more irritable, more on edge, more prone to catastrophic thinking. That's not your imagination. That's your amygdala running unchecked because your prefrontal cortex didn't get the restoration it needed. E.T. Ben Simon, also working with Walker at Berkeley, published research in 2019 and 2020 showing that even one night of sleep deprivation triggered a significant increase in anxiety levels in otherwise healthy participants, and that the degree of deep NREM sleep loss specifically predicted the magnitude of anxiety the following day. Slow-wave sleep, it turns out, has a particular role in calming the brain's emotional circuits. When you lose it, anxiety fills the gap. So let's come back to PTSD for a moment, because this is where the relationship between sleep and mental health becomes especially heartbreaking. Post-traumatic stress disorder is characterized by intrusive re-experiencing of traumatic memories, flashbacks, nightmares, a sense that the trauma is happening again right now. And one of the most consistent findings in PTSD research is that sleep is severely disrupted. Nightmares are a hallmark symptom, but they're not just ordinary bad dreams. They're often exact or near-exact replays of the traumatic event, which is unusual. Normal dreaming, even when it incorporates elements of waking experience, tends to be associative, fragmented, remixed. PTSD nightmares are more like recordings stuck on repeat. Here's what researchers began to suspect. Maybe the nightmares aren't just a symptom of PTSD. Maybe the sleep disruption is actually preventing the trauma from being processed. Remember, REM sleep is supposed to replay emotional memories in a low norepinephrine environment, stripping away the raw emotional intensity. But in PTSD, something seems to go wrong with that process. Murray Raskind, a psychiatrist at the VA Puget Sound, proposed that elevated norepinephrine during sleep, which is abnormal because norepinephrine is supposed to be suppressed during REM, might be preventing the normal emotional processing from occurring. The memory gets replayed, but in a high-stress neurochemical bath. So instead of being diffused, it gets re-traumatized. The person wakes up more distressed, not less, and the cycle perpetuates. Raskin tested this idea with a drug called prazosin, which blocks a specific type of norepinephrine receptor, and in clinical trials with combat veterans, prazosin significantly reduced trauma nightmares and improved overall PTSD symptoms. It didn't erase the memories. It seemed to allow the brain's natural emotional processing during sleep to actually function again. The results were compelling enough that prazosin became widely used in VA settings, though later, larger trials produced more mixed results, and the story is still evolving. but the underlying principle that fixing the sleep disruption can help resolve the psychiatric condition, not just the other way around, that principle has held up remarkably well across multiple lines of evidence. And this bidirectional relationship extends beyond depression, anxiety, and PTSD. Researchers have found sleep disruption implicated in bipolar disorder, where sleep loss can trigger manic episodes, in schizophrenia, where sleep spindle deficits, those stage 2 bursts we talked about earlier, are consistently reduced, possibly contributing to the cognitive fragmentation characteristic of the illness. In ADHD, where sleep-disordered breathing in children can produce symptoms that are virtually indistinguishable from attention deficit disorder, leading to misdiagnosis and stimulant medication, when the real problem is that the child can't breathe properly at night. That last point deserves a moment, because it circles back beautifully to what we were discussing about obstructive sleep apnea. Karen Boenick at the Albert Einstein College of Medicine published a study in 2012 following over 11,000 children and found that those with sleep-disordered breathing were 40 to 100% more likely to develop behavioral problems resembling ADHD by age 7. Think about that. A child who snores heavily, who has enlarged tonsils or adenoids, who experiences repeated microarousals all night long. That child is not getting the deep, slow-wave sleep, or the REM sleep their developing brain desperately needs. They show up at school exhausted, unable to concentrate, impulsive, emotionally dysregulated. And they get diagnosed with a behavioral disorder. In some of these cases, not all, but a meaningful proportion, treating the airway obstruction, sometimes with a simple adenotonsillectomy, resolves the behavioral symptoms. The child didn't have ADHD. The child had a sleep disorder. This is why the reconceptualization of sleep's role in mental health matters so much. It's not academic. It changes diagnoses. It changes treatments. It changes lives. And it raises a deeply uncomfortable question about modern society, which is this: if inadequate sleep contributes to depression, anxiety, emotional dysregulation, impaired judgment, and cognitive decline, and if we live in a society that systematically undervalues sleep, that glorifies busyness, that lights up our nights with screens emitting precisely the wavelength of light most disruptive to our circadian rhythm, then how much of our mental health crisis is, at least in part, a sleep crisis? That's not a rhetorical question. Researchers are genuinely asking it, and the answers, while still incomplete, are sobering. The CDC has called insufficient sleep a public health epidemic. The American Academy of Pediatrics pushed successfully in many districts to delay school start times for adolescents because teenagers have a biologically shifted circadian rhythm. Their melatonin onset is later. Their natural wake time is later. And forcing them into classrooms at 7:30 in the morning is essentially imposing chronic jet lag on an entire population during one of the most critical periods of brain development. When the Seattle School District pushed start times from 7:50 to 8:45 in 2016, researchers at the University of Washington found that students got 34 more minutes of sleep per night, and their grades improved by an average of 4.5%. Attendance improved. Tardiness dropped. 34 minutes. That's all it took. And this gets at something fundamental about sleep that we've been circling around throughout this whole journey, which is that sleep is not a luxury. It's not optional. It's not something you can shortchange indefinitely without consequences. It is a biological process as essential as breathing, as non-negotiable as a heartbeat. and it performs functions that nothing else in your waking life can replicate. No amount of caffeine, no meditation app, no supplement can substitute for what your brain and body accomplish during a full night of properly architected sleep. And yet, we treat it as negotiable. We brag about how little we need. We design work schedules and school schedules and social norms around the assumption that sleep is flexible, compressible, something to be optimized away. And the consequences show up everywhere, in our health, in our mood, in our relationships, in our ability to think clearly and make good decisions. They show up in highway accident statistics and medical error rates, and the slow, invisible accumulation of amyloid plaques in the brains of people who spent decades sleeping 5 or 6 hours a night. Which brings us to something that's been developing quietly in sleep science over the past decade or so, and it has to do with the question of individual differences, because not everyone seems to need the same amount of sleep, and there's been a long-running debate about whether some people are genuinely wired to thrive on less. And the answer turns out to be genuinely fascinating, because it sits right at the intersection of genetics, neuroscience, and the stories we tell ourselves about who we are. So here's what we know. In 2009, a research team led by Yinghui Fu at the University of California, San Francisco, identified a mutation in a gene called DEC2, also known as BHLHE41. They found a mother-daughter pair who consistently slept about 6.25 hours per night, naturally, without an alarm, without feeling tired during the day, without any of the cognitive or health consequences you'd expect from chronic short sleep. And when Fu's team engineered the same mutation into mice, those mice slept less too, but performed just as well on memory and learning tasks as their normal sleeping counterparts. This was a real, measurable genetic difference. Not willpower, not habit, not caffeine dependency masquerading as resilience. Their brains genuinely needed less sleep to accomplish the same restorative work. Since then, Fu and her colleagues have identified a handful of other genes associated with natural short sleep. There's a mutation in ADRB1, the beta-1 adrenergic receptor gene, published in 2019. There's one in NPSR1, the neuropeptide S receptor gene. Each of these mutations is rare. We're talking about a tiny fraction of the population. The estimates vary, but most researchers put the number of true genetic short sleepers at well under 1%. Maybe a fraction of a fraction. And here's the thing that matters. For every person who genuinely carries one of these mutations, there are probably hundreds or thousands who believe they're fine on 5 hours, but are actually walking around in a state of chronic partial sleep deprivation that they've simply normalized. This is one of the trickiest aspects of sleep science, and it's worth sitting with for a moment. When you're chronically underslept, one of the first things that degrades is your ability to accurately judge how impaired you are. There was a landmark study by Hans von Dongen and David Dinges at the University of Pennsylvania, published in 2003, where they took healthy adults and restricted them to either 4, 6, or 8 hours of sleep per night for 14 consecutive nights. The people sleeping 4 hours showed cognitive impairment equivalent to going 2 full nights without any sleep at all. The 6-hour group was nearly as bad after 2 weeks. Their reaction times, their attention, their working memory, all measurably degraded. But here's the kicker. When you asked them how sleepy they felt, their subjective ratings plateaued after just a few days. They stopped feeling worse. They adapted to feeling bad. They recalibrated their internal baseline so that impaired became their new normal, and they genuinely believed they were functioning fine. Think about what that means. It means that the person who tells you, with complete sincerity, that they do great on 5 hours of sleep, They might be telling you the truth about how they feel. But feeling fine and being fine are two very different things. Their reaction time might be slower. Their emotional regulation might be compromised. Their immune system might be quietly struggling. Their brain might be accumulating the kind of metabolic waste we talked about earlier, the stuff the glymphatic system is supposed to clear during deep sleep. And they'd never know, because the impairment is invisible to the impaired. So when we talk about individual differences in sleep need, we have to hold two truths at once. Yes, there is genuine genetic variation. Some people really do need a little less, some need a little more. The bell curve for optimal sleep duration in adults probably centers somewhere around 7 to 8 hours, with a range that extends from maybe 6.5 to 9 hours for most people. That's real variation, and it matters. But the cultural narrative that celebrates short sleep, the CEO who sleeps 4 hours, the entrepreneur who treats rest as weakness, the surgeon who brags about 36-hour shifts, that narrative is not supported by the science. For the vast majority of people, consistently sleeping less than 7 hours is associated with measurable harm. Not because they're weak, because they're human. And this brings us to something that feels like a natural place to land as we come toward the end of this whole journey through the science of sleep, which is the question of what we do with all of this knowledge. Because we know so much more now than we did even 20 years ago. We understand the architecture, the slow waves, the spindles, the REM cycles, the 90-minute ultradian rhythm that carries you through the night like a tide. We understand the chemistry, adenosine building its pressure, melatonin signaling the darkness, cortisol rising with the dawn. We understand the circadian clock, those tiny clusters of neurons in the suprachiasmatic nucleus keeping time with a precision that still amazes me, synchronized to the rotation of the planet by photons hitting specialized cells in the back of your eye. We understand what sleep does for memory, how it replays and consolidates and integrates, how it files away the important things and lets the noise fade. We understand what it does for emotional health, how REM sleep strips the sharp edge off difficult experiences, how losing it makes the amygdala run hot and the prefrontal cortex go quiet. We understand the glymphatic system, those channels that open up during deep sleep to wash away the day's metabolic debris. We understand the immune connections, the metabolic connections, the cardiovascular connections. We know all of this, and yet, the gap between what sleep science has discovered and how society actually treats sleep remains enormous. And bridging that gap is in some ways the great unfinished project of this entire field. It's not enough to understand the mechanisms. The mechanisms have to change behavior, and behavior has to change culture, and culture has to change policy. And that chain, from lab bench to lived experience, is long and slow and full of friction. But there are signs of movement. We talked about the Seattle School District, and they're not alone. California passed a law in 2019, Senate Bill 328, mandating that middle schools start no earlier than 8 o'clock and high schools no earlier than 8:30. It was the first statewide law of its kind in the United States, and it went into effect in 2022. The American Academy of Pediatrics had been recommending later start times since 2014, but it took years of advocacy, years of presenting the data, years of parents and teachers and sleep researchers pushing against the inertia of bus schedules and sports practices and the simple human resistance to changing the way things have always been done. In medicine, there's been a slow reckoning with the culture of sleep deprivation that has long defined residency training. The old model, 100-hour weeks, 36-hour shifts, the idea that exhaustion was a rite of passage. That model was challenged after the death of Libby Zion in 1984, an 18-year-old woman who died at a New York hospital while being cared for by overworked, underslept residents. Her father, Sidney Zion, was a journalist, and he turned his grief into a crusade that eventually led to the Bell Commission regulations in New York State, limiting resident work hours. The Accreditation Council for Graduate Medical Education followed with national limits in 2003, capping resident duty hours at 80 per week and continuous shifts at 30 hours. Even those numbers sound staggering, don't they? 80 hours a week, 30 hours straight, and there was, and still is, genuine debate within medicine about whether these limits help or hurt, whether shorter shifts introduce more handoff errors, whether the old way really did produce tougher, more competent doctors. But the sleep science is pretty clear. After about 16 hours of continuous wakefulness, cognitive performance starts to decline. After 24 hours, it's equivalent to a blood alcohol level of 0.1%, legally drunk in every state. The idea that we'd want someone in that state making decisions about medication dosages or surgical incisions is, when you step back and think about it, genuinely alarming. And then there's the workplace more broadly. Some companies have started to take sleep seriously, not as a wellness perk, but as a performance variable. There are organizations experimenting with nap rooms, with flexible schedules, with policies that discourage after-hours email. The research supports it. A study by researchers at Harvard estimated that sleep deprivation costs the U.S. economy over $60 billion a year in lost productivity. Not because people are sleeping at their desks, but because they're showing up underslept and making worse decisions, being less creative, getting sick more often, having more accidents. The economic argument for sleep is actually quite strong, which matters because sometimes the economic argument reaches people that the health argument doesn't. But I think the most important shift isn't happening at the policy level or the corporate level. It's happening at the individual level, in the quiet recognition that sleep is not something to be conquered or optimized away, but something to be respected. And maybe that sounds soft or obvious, but it runs against a deep cultural current, especially in places like the United States, where productivity is practically a moral value, where busyness is worn as a badge, where the phrase, "I'll sleep when I'm dead," is said without irony, despite being, as Matthew Walker has pointed out, tragically self-fulfilling. The science we've walked through tonight tells a different story. It tells us that sleep is not passive. It's not the absence of wakefulness. It is an active, elaborate, precisely orchestrated biological process during which your brain and body do things they cannot do at any other time. Your neurons replay the day's experiences and strengthen the connections that matter. Your cerebrospinal fluid pulses through channels that only open when you're deeply asleep, carrying away waste products that would otherwise accumulate and damage your brain. Your immune system calibrates itself, building the defenses you'll need tomorrow. Your emotional brain processes the difficult things you experience today so that tomorrow they'll feel a little less raw, a little more manageable. Growth hormone surges through your body, repairing tissue, building bone, maintaining muscle. Your cardiovascular system gets a break it desperately needs, your blood pressure dropping, your heart rate slowing, your blood vessels relaxing into a state of recovery that protects them from the relentless demands of waking life. All of this is happening right now, or it will be soon, as you drift off. And that's the beautiful thing about understanding sleep. It doesn't require you to do anything extra. You don't have to try harder. You don't have to perform. The machinery is already there, built into you by hundreds of millions of years of evolution, refined and tested and optimized by every generation of every species that has ever slept. All you have to do is let it happen. And I think there's something deeply comforting about that. In a world that constantly asks you to do more, to be more, to optimize and hustle and grind, sleep asks nothing of you except to stop, to let go, to trust that your body knows what it's doing. Because it does. It has been doing this since before you were born, since you were a fetus spending most of your time in REM sleep, building the neural architecture that would become your mind. It will keep doing it tonight, cycling through its stages with the same quiet reliability as the turning of the earth, the same rhythm that Demiron's mimosa plant followed in that dark closet 300 years ago, the same ancient clock that ticks in the cells of every living thing. So as you lie here, wherever you are, whatever kind of day you had, whatever is waiting for you tomorrow, know that your brain is about to do something remarkable. It's going to take everything you experienced today and decide what matters. It's going to strengthen some memories and let others fade. It's going to process emotions you might not even be consciously aware of. It's going to clean house, literally flushing out the molecular debris of a day spent thinking and feeling and being alive. And it's going to do all of this without any effort from you at all. The adenosine that's been building in your brain all day is doing its job right now, pressing gently on those sleep-promoting neurons, making your eyelids heavy, making the world feel soft and distant. Your suprachiasmatic nucleus registered the fading light hours ago and sent the signal to your pineal gland, and melatonin has been quietly rising in your bloodstream, telling every cell in your body that it's time. Your body temperature is dropping just slightly, just enough. Everything is converging toward the same destination. You've spent this time learning about something you do every single night, something so fundamental that life itself might not exist without it, and now you get to experience it firsthand. The slow waves will come first, those long, rolling electrical rhythms that wash across your cortex like waves on a shore, and with them the deep, restorative sleep that clears your mind and heals your body. Then the cycles will begin, carrying you through lighter stages, through bursts of sleep spindles that are locking in everything you learned today, and eventually into REM, where your brain will light up almost like it's awake, dreaming its strange and necessary dreams while your body lies perfectly still, held safe by the ancient mechanism of atonia. And then it will do it all again. And again. Each cycle a little different from the last, the early ones rich in deep, slow-wave sleep, the later ones longer in REM. The whole night a carefully composed sequence that your brain has been performing since the very first night of your life. You don't need to think about any of it. You don't need to remember any of it. It's all taken care of. The only thing left to do is the easiest thing in the world, which is nothing at all. Just breathe. Just be here. Just let the warmth of wherever you are hold you, and let your thoughts soften, and let the quiet take over. Everything else can wait until morning. Right now, this is enough. This is exactly where you're supposed to be.